So I'm Rita, and I'd first like to thank the organizers for giving me this opportunity to speak, and I thank the audience for coming, even though we're all freezing, I think. So I am a microbiologist at WVU, and I'm housed in the Department of Biology. And I have a disclaimer. In 4th grade, I won my spelling bee. So I do know how to spell. It was just my little way of being, cute and catchy, I guess. So today we're going to talk about microbes. And I have 3 aims in this talk. The first aim is to provide you with a little bit of a historical perspective on what microbes are, where we find them, when do we find them. Then I'm going into one of the hottest areas in microbiology right now, and that's the microbiota. So think of this. These microbiota are microbial symbioses, right? So microbiota and in particular host-associated microbiota, are the microbes that are living on or within an animal. So we have different microbiotas. And so I'm going to use humans as an example. And I'm going to tell you how they're important for our biology. And then I'm going to tell you how we get microbes, okay? And then I'm going to go into one area that's been greatly accelerated through studying microbiotas. And that's the understanding of vectors. And this guy is what I study in my lab, so it's the Teti fly, right? And it's actually a cute insect, so it's not as it's not as disturbing looking as this, but the diseases it transmits is very devastating, right? And obviously it's not true to size right here, right? Because we'd all be in trouble. So microbes are very much among us. First microbe hunter was in the 1600s, and that was Leeuwenhoek, and he discovered the first microbes. And why he did that was he created the first lenses that made it possible to see microbes, right? So microbes are any organism that we need special lenses that we can't see with the naked eye. Right? So Leeuwenhoek back then, he called them little animals. And right now, we consider these little animals the microbes. And so there's stuff like bacteria and viruses and protozoa. They're everywhere. Okay? So they're in our oceans, they're on our soil, they're on our bodies, they're within our bodies. They're even found in the most inhospitable places on Earth. So they were the first life forms on Earth, and then they flourished ever since. They're plentiful, okay? We're talking mind-boggling numbers. My favorite example is if you pick up a teaspoon of soil, on average, that teaspoon of soil will contain 1 billion microbes. To put that into perspective, that's the amount of individuals that are currently living in Africa today, and that just 1 teaspoon of soil. So very plentiful. And they get this unfair bad reputation, okay? So this morning, Judith talked about the bad side of the microbes. You learned about the HIV, the hepatitis C virus. And there certainly are bad microbes out there. And I think that this perception of all microbes being bad really started with Louis Pasteur's germ theory, right? So he basically, and he was spot on, he said, well, infectious diseases are caused by microbes, and that's completely true. But I think it pushed the general public to think of microbes as with an ick factor. In fact, most microbes don't do anything to us or are quite beneficial to us. Okay, so today we recognize about 1,400 different human pathogens. So those are the bad guys. And that's a tiny, tiny, tiny sliver of the microbial diversity that's out there. We're talking about less than 0.001% of the microbial diversity are actually bad for us. And they're unculturable for the most part, okay? Meaning we can't grow them up in defined media, okay? So we know they're there, but we are not able to grow them up. And that presented a major crux in microbiology, okay? And I was very fortunate because I started getting to microbiology at right about a really good time. And why is that I started getting into microbiology when the sequencing revolution started. Okay, so this was in the 1990s, right? I just got my master's, getting into my Ph.D. And the sequencing technology just blew up, right? Revolutionized medicine, revolutionized biology. Because how it did that is that we no longer need to grow up these microbes to try to understand them. These sequencing technology, and now there's been multiple generations of sequencing technologies, they gave us the ability to learn exactly who's there, what they're doing without needing to grow them up. Okay, we just need to isolate their DNA and RNA, and if you're just like a kind of okay cook, you can do that. It's just all following basically recipes that scientists call protocols. It's the same thing. Okay, so microbiotas. I'm going to use humans as an example. There was a great mystery that was presented in 2001. Okay, 2001 was a major advancement in science. Our genome got sequenced and annotated. So basically, you know exactly what we're capable of genetically. All right? And the big surprise was, it was astounding. We only have about 25,000 genes in our genome. Right? So when these guys were sequencing the genome, they expected so many more genes to be discovered. Only 25,000. And that made people think, wow, how are we functionally capable of doing all these activities, breaking down all these metabolites, right? Doing all these molecular mechanisms with just 25,000 genes? How is that even possible, right? And as an aside, there's stuff out there, plants, there's amoeba that have many more genes than we do, okay? So the human genome consists of about 25,000 genes. So then the great mystery, well, how are we capable of doing what we can do? I think we started learning more about this when the inception of the Human Microbiome Project, and that's funded by the National Institutes of Health, and Europe has its own version as well. And so what the Human Microbiome Project basically unraveled, right, is that if you compare the number of human cells, so the number of our cells, to the number of microbial cells that are living on or within us, let me remind you, that's the microbiota, right? We're only about 43% human. 57% of us is microbial, and most of that's dominated by bacteria, okay? And if you actually break it down to the gene level, right? So remember I said we have about 25,000 genes in our genome? Well, the microbes, part of our microbiota, are giving us about 2 million to 20 million genes. So if you take that ratio, we're about 1% human. So scientists, microbiologists have started asking the question, are we more microbial than human? And who's controlling who? So now you're probably like, oh my gosh, they're really important. How do we get microbes, right? I hope that's where I led you. How we get microbes, how they're transmitted. Thank you, Mom, right? You gotta thank your mom for your mitochondria, because that's where we get our mitochondria, and we get our microbes in the beginning from our moms. We know right off the bat, right, that babies that are born C-section versus natural birth have very different microbiotas. Furthermore, if they're formula fed versus nursing, they'll also have very different microbiotas. Okay? Let me tell you this astounding fact, in that breast milk contains sugars, they're known as oligosaccharides. And we know, the pediatricians know, that these babies, the neonates, newborn babies, are unable to process these sugars. These sugars are there, made by our bodies to promote the good bacteria to establish. So here we're making sugars, one species, us, making these sugars for another species to establish, okay? So there's lots of money actually nowadays trying to figure out how to make formula, right, more reminiscent of breast milk. Okay, so my lab has discovered that animals that live within the same location tend to have more similar microbiotas than animals that don't live in the same location. Okay, this can be exemplified by my brothers and I, okay? So here, right, it's exemplified by my brothers and I. As children, we had more similar microbiota to each other than to children that lived outside of our house. Furthermore, right, so this is my son, and so I have 2 kids, well I have 3 kids. My son, and this is my dog. I call my dog my kid too, right? So I could tell you that Microbiotas between family members tend to be greater if there are pets involved, particularly dogs, right? So, and for the dog lovers out there, we do share some of our microbiota with our dogs as well, okay? So you can actually figure out people and who their dogs are by matching up microbiota. So research in my lab has already determined that the existing microbiota can also influence who's going to come on board. Okay, so this should impact you, right? Like this should, wow, well, C-section babies versus natural birth already start from the beginning. That means that they're going to be influential on who can come on board later, right? And so research in my lab has shown that, you know, these later colonizers are going to be determined by who's already there. And how they do this is through competition, right? Competition, cooperation. So they basically throw up this biological equivalent of no vacancy if they don't want it there, right? Through this enhanced competition. So now I should have led you down to this question: how much of an impact does our microbiota really have? So this is an experiment that really influenced me. This is really early on in my career. I learned about this study, and I'm going to guide you through this experiment. Okay, so this is a mouse experiment, and what they did is they have pregnant mice, and they surgically remove the pups. Okay, so they surgically remove the pups, and so the pups are germ-free. They're not exposed to any microbes. And then they're rearing the mice in germ-free conditions, okay? So it costs a lot of money to raise these mice lines. And then, now we have two brothers, right? So two mice, they're brothers, you know, pretty similar, high genetic identity between them, genetic similarity. They're being reared in germ-free conditions. And now they're receiving chow, so they have to get fed. They're eating the same amount of food. The food is sterilized, so you can't think that microbes are coming on board that way. The only difference is that I'm going to show you the picture of one mouse. He's exposed to the microbiota of a lean mouse. This one is exposed to the microbiota of an obese mouse, okay? So obesity does have a microbial component to it, and there's lots of interest trying to understand how microbiota can be influenced, like how we can distort and manipulate the microbiota to better promote better health. And there's also, you know, the imbalances and distortions in the microbiota, not only in who's there, but what they're doing, has also been implicated in a wide range of diseases. Okay, and this is just one short afternoon. I'm researching NIH's PubMed library, and this is what I've been coming up with. So these diseases, there's research out there. It seems that there's a microbial component to the onset of these diseases. Okay, let's switch over to vectors, right? So the microbiotas, I hope I convinced you. You now know what they are. We now know how we get them. You know how they're important. One area of research that's been greatly enhanced by studying microbiota is vector biology. Okay? So vectors are typically insects and ticks that transmit disease. And these diseases that they cause are due to worms, protozoa, bacteria, viruses. There's often very tight coupling between the vector and the pathogen, and that's due to a tremendous amount of coevolution and adaptation that they've done. So not every mosquito can transmit every disease out there. It's very tight species-to-species interactions. Okay, so 17% of all infectious diseases are vector-borne, and 25% of all mortality in the world is due to infectious diseases. So huge morbidity and mortality associated with vector-borne diseases. So the disease that's receiving probably the most amount of recognition right now, a vector-borne disease, is Zika. So let me tell you a little bit about Zika. So Zika was first isolated in the Zika forest in the 1940s in Uganda. and it was infecting monkeys, non-human primates. And since that identification in the 1940s, we saw sporadic outbreaks, and these were mostly confined to parts of Africa. And then we fast forward to 2007. In 2007, on Yap Island, so this is in the Federated Micronesia states, what we're seeing in Yap Island is that there's a very high amount of infection. Like the majority of people are getting infected on that island. And what was interesting is that there is no associated pathology with infection. People are getting infected, but not really getting sick. If we fast forward to French Polynesia in 2014, we're seeing the same high rates of infection, right? We're seeing the same high rates of infection. The majority of people that are living there are getting infected. Now we're starting to see some pathologies associated with it. So something happened, a switch happened. And most of the pathology is in the form of Guillain-Barré syndrome. So this is a muscle weakness that's due to damage of the peripheral nerves. You fast forward that now 1 year to Brazil, and that's when all these microcephaly cases start being reported. So this is those newborns that are their moms are infected with Zika virus. The newborns, they're born, they have the, reduced skull and brain development. And it's associated with really significant developmental impediments. So we're seeing those reports. Then in 2016, it's in the US, right? So it's here. And not only is it here, but there's local transmission, and it's been reported in at least 2 states. Local transmission means the endemic mosquitoes, the mosquitoes that are naturally circulating in our environment, are now passing Zika virus around. And that's been reported in Texas and Florida. And this year, I think it's going to increase even further. So one way, and this is not work that's been done in my lab, but it's, I think, a very nice, clever way, savvy way to show you how the microbiota can be used to control these vector-borne diseases is through this strategy. So this is Aedes aegypti. So these are the mosquitoes that, this is the main vector for Zika virus. And Aedes aegypti, what we know is that it can become infected with this Wolbachia bacteria. It's a part of its microbiota, okay? And so what we have here is one mosquito cell, and there's at least 3 different Wolbachia infections. So Wolbachia is a bacteria, it's part of its microbiota. There's 3 important points to make about Wolbachia. 1, it can't infect us. It's never known to infect vertebrates. It only infects invertebrates, okay? Very evolutionarily distant from us. 2 is that any mosquito that's infected with Zika virus is unable, any mosquito, that's infected with Wolbachia is unable to be infected with Zika virus. So if it has Wolbachia in its microbiota, it can't have Zika virus. Zika virus can't establish within that mosquito. And 3, just as important, is that when we know that mosquitoes that have this Wolbachia infection tend to have a reproductive advantage over mosquitoes that do not have this Wolbachia infection. Okay, so here's the strategy, and I'm going to go over it briefly. Is in the lab, they rear Aedes aegypti. It's super easy to rear mosquitoes, have lots of them. They reproduce like crazy, so they're in the lab, and you infect them with Wolbachia. And I have to say, this is being currently used. In parts of Florida, this is being used, and in other parts of the world as well. And so we have Aedes aegypti in the lab. We infect them with Wolbachia, and then we get them into high numbers, and they are now released into the field. Okay, so this schematic is telling you that the black mosquitoes are mosquitoes without Wolbachia. The green mosquitoes represent those mosquitoes that are infected with Wolbachia. And what you see is, in the beginning, the wild, of course, mosquitoes don't have any Wolbachia. And then there's the release period, right? And through time, you're seeing that the green start outnumbering the black and eventually totally replace the black mosquitoes, those mosquitoes that don't have Wolbachia infection. And remember that this happens because of that reproductive advantage. These female mosquitoes are putting out more mosquitoes than those uninfected mosquitoes, okay? And remember, so now we're going to have a population here of all these green mosquitoes that have Wolbachia, and they're unable to transmit Zika virus. Thereby, Zika virus prevalence in the field is also going to get reduced. Okay, so this is a really nice, savvy way that you can use the microbiota to try to decrease vector disease. So in my lab, one of the animal models that we work on is the Teti fly. So Teti flies are very important medically because they're the obligate vector of sleeping sickness, also known as human African trypanosomiasis. This is one of the most neglected diseases in the world. This has been identified by Doctors Without Borders as a disease that's in much need of some attention and some control. It also has pretty big agricultural significance because it causes a wasting disease in cattle known as Nagana. And if you've been to Africa, you know that a lot of Africa is very lush, and actually establishment and development of these areas is inhibited, and one of the big reasons is because of the tsetse fly prevalence in those areas. So here is an infected patient. So you take we smeared on a glass slide, and these are our red blood cells, and these are the trypanosomes, right? So they're extracellular. And they're solely confined to sub-Saharan Africa. And they're quite unusual with insects. So I love insects ever since I was a little kid, right? Fascinated. Most insects, and I'll try to teach anybody about insects anytime. So most insects, if they are blood feeders, it's the female that will be the blood feeder, right? And most of the time she needs that for egg development. We see that to be the case, for example, in mosquitoes. The tsetse flies are quite unusual in that both the males and the females will take blood meals. So that has a huge epidemiological significance in the field because now both sexes can transmit the disease. Tsetse flies are quite unusual as well because they have a very low reproduction rate, okay? And this picture indicates why that low reproduction rate occurs. So here is this female fly. And she's fertilized fly, and she just deposited that. That's a larva, okay? So the majority of the development of the larva occurs in utero, very similar to humans, right? So that would be pretty much if a human gave birth to a 13-year-old. That's how much development happens within the mother's uterus. And oftentimes, that larva weighs more than the mom itself. So right there, it should be an indicator of how high that maternal investment is, right? And because she invests so much in her progeny, she can only have about 6 to 7 offspring per lifespan. And so to us, that represents a really nice Achilles heel to try to impede and really try to bring down those population numbers. And we're very fortunate at WVU. We have one of the few in the world colonies of tsetse flies in my lab. And so that has allowed us to answer so many great questions to do with microbiota, being able to extrapolate those to other animal models, but also apply to a very medically significant vector. So we've characterized the tsetse microbiota to be very simple. And we know that that's true. It's only comprised of a couple different bacteria. And we know that that's true not just in our colony flies, but when we go out in the field, we see the same thing. Very simple complexity microbiota. I'm going to show you in a couple slides how they're very crucial for tsetse biology. And that's really nice because it presents a way that if you could impede the microbiota in some sense, it could act as a novel control mechanism for the diseases that this vector transmits. So it's another tool in our arsenal for tsetse control. Okay, so at this point, you should be asking, okay, now we have this tsetse fly. She's telling me about this microbiota, and I know there's trypanosomes that come into the story. When do they all meet up? And so this is what happens. This is a cartoon that I'm going to show you when you get really creative undergrads in your lab. And I think that they're creative, and then they become very frustrated with how their professor gives them a life cycle, and then they're like, oh, and they're like, Professor Rio, we can do it much better, right? So this is something that a undergrad, he's now in med school, Keegan Guffey created for me to show you how where the trypanosomes and microbiota all meet up in the tsetse fly. And so you'll learn a little bit about vector biology, okay, too. Okay, so the trypanosome and tsetse life cycle starts when the tsetse fly obtains an infected blood meal. So this person's infected, and so the tsetse fly is taking up trypamastogotes in the form of the slenders and the stumpies. The stumpies are the ones that are going to go into the midgut of the fly and proliferate. So the fly now takes off, it's not on the host anymore, and the trypanosomes require microbiota to breach into this space in the fly where they can developmentally differentiate, and they replicate like crazy, and they head towards the salivary glands by way of another member of the microbiota known Wigglesworthia. It's very important for tsetse fly biology. That's why it's got the white coat on. And then it heads up to the tsetse fly salivary glands. It attaches, recombines, and the cycle is over when a new naive host gets a blood meal. No, it takes like 12 days, just 12 days in the fly for it to do that whole life cycle, right? So really intricate biology that I was talking about, coevolution between the two partners. So in my lab, we've discovered that the tsetse microbiota, like for humans, is crucial for providing essential nutrients, okay? And what my lab's discovered is B vitamins. B vitamins are being provided by the microbiota. We've sequenced genomes, we've seen which genes are being turned on, and these are B vitamins. B vitamins, when you do like a metabolomic profiling of blood is deficient in B vitamins. If we're talking about flies that just take strict blood meals, they're going to have to get these essential vitamins in some other way. Animals are incapable of making vitamins. So these guys, the microbiota, is making those lacking B vitamins. And to us, that represents a really nice target area to try to impede the tsetse fly biology by targeting these pathways. And so we did exactly that. So here's the fly, right? It's really beautiful. Let me tell you, so this is a female, right? And she's got the larva right here. She's about to give birth, right? And so she also just took a blood meal. So they become really fat. And they're just like, in the lab, people love coming to visit them and see how we feed and all that. So it's never an issue to get undergrads in my lab, right? So what we did here is, in this experiment, we took advantage of that B vitamins that the microbiota is making, and we said, well, what would happen if we inhibit that B vitamin? So we did this enzymatic inhibition study, and we saw that the larvae that had the microbiota blocked in B vitamin provisioning, they were significantly smaller than the normal flies that were not blocked, right? And then we could rescue back this effect just by simply putting the B vitamins into the blood, which makes it indicate specifically, yes, we were blocking that B vitamin biosynthesis by the microbiota, and that led to these impediments in development, okay? So this is, in the future, could very much have a lot of application towards reducing the tsetse population. So I have to say that I'm a selfish person, too. And one of the reasons why I went into vector biology specifically is because I got to travel to really cool places. And so my research group, we get to go to fields. I mean, it's not all about just sticking in the lab and working with your colony flies, although it's really nice to have that tool and those resources, but getting out in the field. And getting out in the field, this is a picture that I have, thousands of these pictures because this is exactly what it looks like all the time, right? So these are my companions in the field. This is in Kenya. These are the mountains of Tanzania. So the vector biology gives you so many great opportunities for global engagement. And not only do we, know, like the research group gets exposure to the animals and the wildlife, we get exposure to the culture. But by far, my favorite part of traveling is meeting the kids, right? Because the children are just, just joyful. I mean, everywhere in the world, they're just joyful and really great to learn about them. Here's some children from the Maasai tribe that we got to hang out with for an afternoon. So this is in the Kenyan highlands. And this little guy was probably one of my favorite because he laughed at all my jokes, which is almost impossible, right, to ever find. But this kid was great. So what we did and what we've been doing recently is we've been taking that tremendous sequencing technology that I'm, applauded in the beginning and applying it to the field, okay? And so for the next two slides, I'm sorry, they're kind of data heavy, but I'm gonna walk you through them a little. So what we do is we go out in the field, we take our samples, we're dissecting these flies there's no electricity out there. It's all solar. So when the sun goes up, you start working. When the sun goes down, you stop working. So we're dissecting these flies, we're putting them in special chemicals, and then shipping them back home, right? And then using this sequencing technology to tell us what's happening in the field. And what's happening in the field is really awesome, because what we're seeing, and so these, you could see here on the x-axis, this represents different genes in the microbiota. And this, on the y-axis, you basically just can interpret it as how high is a gene being turned on, okay? So where genes that are being turned on means that they're super important for the biology. And so a lot of the genes that we're seeing that are turned on are in this B vitamin synthesis. So this is a real deal. We're seeing this in the field, that this microbiota and the B vitamins that they're providing are very important to the field flies. Furthermore, when we look, right, so now we take these tissues that the microbiota, the trypanosomes are in within the Teti fly, we could see through this great sequencing technology, what else are they doing? We're seeing that the majority of genes that are being turned on all fall, like majority of them fall in metabolism. So there's so much more to discover, so many more genes and pathways that can maybe not only increase our understanding of the Teti fly biology, the trypanosomes, and how Tetis are vector competent for these diseases, but they also provide nice ways that we can potentially apply for control, okay? Lots of molecules being transported between the Teti fly and the microbiota, right? And I made it blue and gold for you guys. So just to recap on my talk, microbes, they're everywhere, right? And hopefully that's not such a bad thing for you guys to think about. It's not such an ick factor anymore. I think we should honor thy microbiota. Has a lot of important biological significance. And I don't know how many of you caught Judith's talk in the morning about the opioid addiction and how rampant opioid use is in West Virginia. West Virginia also gets the honor of the highest level of antibiotic use, right? Antibiotics are going to distort microbiota, and that's going to create significant issues as well. So honor thy microbiota. You can influence, there's different health styles and choices that you can make to try to promote a healthier microbiota. And manipulation of the microbiota offers new benefits for society. And I showed you how, I tried to grab you with the human side of it. I was going to apply it, I'm applying it to the vector side of it because I love the insects, right? But you could think of it, other animals can also benefit from this. And I have colleagues that are trying to do some similar stuff with honeybees, right? And trying to promote their colonies out there in the United States. So 2015, 2016, or 2014, 2015, 40% of the US bee colonies were had decreased. And these are 1 out of every 3 teaspoons of food that we get are pollinated by a bee. So huge importance to us. Okay, so I am, admittedly, a very small piece of this puzzle. But I have to say, like Carolyn said, right, I'm a first-generation college student. So this is what happens when people support people like me, right, is that I can do good things. So I'm a little piece of this puzzle. The engines are the undergrads. The grad students that I've had in my lab, it's been an honor to have them in my lab. They're brilliant, They're motivated, hardworking. And you know, those three, you guys are all successful out there. That's a dangerous combination, right? So they're hardworking, and I think Alexis was the one that said, you know, they're a huge motivator. I mean, I would have to disagree. I think the public is saying, oh yeah, you know, they're crybabies, blah, 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 out there. These guys know how to move things. So I've been privileged to have great members in my lab, great collaborators. West Virginia University, I have to applaud them because when they hire a professor, they take a huge risk, right? So they're basically saying, yeah, I believe in you, here's a pot of money to try to go and create more with that money. And they've been so supportive of me, not just in the beginning of my career, but all throughout. And I've been here, this is my 10th year at WVU. I've had over 50 undergrads go over through my lab, and they've all landed in some kind of professional schools. Sadly, I don't think one of them has stayed in West Virginia. They've all done tremendously great things, it's just the opportunities are not here. And then I definitely have to acknowledge my funding sources. They've been very generous in supporting my work, and that's the National Science Foundation and National Institutes of Health, specifically NIAID. And with that, thank you so much.